So welcome, everybody. It's a great pleasure for me that I have the opportunity to present here our research on developmental toxicity of micro-nanoplastics. And let's just start. So I think I don't have to say to this community that micro-nanoplastics is appearing to be seen as a major concern in pregnancy. So micro-nanoplastics arise from our plastic products, from degradation, or they're already produced in this small size. And they are increasingly accumulating in our environment due to the high biopersistence. And of course, there's still an increase in global plastic production. And pregnant women are of particular concern because they're highly vulnerable, and also the developing fetus. So should we be concerned about micro-nanoplastics? There is some first studies from animals that could indicate that there's maybe a risk for developmental toxicity. But I have to say most of these studies so far are done with polystyrene model plastic particles, because the issue is that we don't at the moment have environmentally relevant micro-nanoplastics in these really small size fraction that we could study the environmentally relevant plastics. However, these studies with these model particles have shown that there could be some concern, as they have seen exposing animals that this can reduce placental and fetal weight, and also have some toxicity to the developing fetuses, like neural tube morphogenesis defects or injury to the fetal thalamus. So we started also to work in this field a bit like more than 20 years ago, and at that point there wasn't really much known about nanoparticles at all. Do nanoparticles cross to a fetus across the placenta barrier? Is this dependent on the properties of the materials? And of course not at all what is the health impact if these things would cross placental barrier. So when we talk about pregnancy, I want to emphasize that we should not just talk about toxicity to the fetus, but there is one organ that is so central in pregnancy, which is the placenta, and sometimes overlooked, because it has so many functions that are crucial for pregnancy, like not only acting as a barrier to protect against toxicants, but also like having exchange functions or immunological, hormonal, endocrine functions or metabolic functions, which are crucial. And with all these important functions in pregnancy, it's very important, easy to understand also that any exposure to toxic substances could be detrimental for fetal development. Another important fact for our research is that the placenta is considered the most species-specific organ. There's tremendous differences between species in structure, morphology, physiology, and pathophysiology. And it's also the organ that we can access after pregnancy from humans. And we in our lab use a variety of different models, mostly based on the human placenta, going from high-throughput models like isolation of primary cells from the placenta up to more sophisticated models, like the most sophisticated gold standard model is the perfusion of an intact placenta. So the overall goal of our research is that we aim to contribute to a better understanding of developmental toxicity of micro-nanoplastics. And we center on placental uptake and translocation, on toxicity on the placenta, and toxicity on the early developing embryo. So when we look at the transport, one of the gold standard models that we can use using human placenta is a placenta perfusion. So you can obtain a placenta immediately after cesarean section. You can recannulate fetal blood vessels, artery and vein, and cannulate the maternal side to re-establish a maternal blood circulation and a fetal blood circulation. And then you can add microparticles, nanoparticles to the maternal side and see if they cross to the fetal side. And you can do that for 6 hours. Afterwards, the organ is slowly coming to the end of life. So we did some very early studies already more than 10 years ago, and we, of course, also used this model, polystyrene micro-nanoplastics, in different sizes. And what you could see is that when you add these particles to the maternal side, you can see that they decrease in the maternal circulation, but they also slowly start to appear on the fetal circulation, indicating that yes, micro-nanoplastic can cross a placenta barrier, and this transport is size-dependent with very low transport for larger particles, but smaller particles seem to cross. But is it only about size? So we tested some other particles with other properties, for instance, carboxylate polystyrene, which has a negative surface charge, and we could see that it's not as simple. So there's also other properties that contribute, and we could see that these carboxylate particles, even if they are really small, they no longer pass the placenta well. So also these properties seem to have an influence. But what we also found is that overall, all the particles, even if they didn't cross, they accumulate in this placental tissue. Our data were then later on confirmed with some epidemiological studies. So in 21, Ragusa's lab showed that microplastics, so they cannot still not detect the nanoplastics, but at least the microplastics is present at the placenta, which of course raised a lot of concerns. Like there was this statement from Antonio saying like it's like a having cyborg baby, no longer composed of human cells, but also a mixture of biological and inorganic identities, which is maybe a bit attention-grabbing, I'm not yet sure. And a year later, there was another group showing that microplastic is also present in meconium, which is the first stool of the unborn kids, so should also indicate that there was a fetal transfer. So what about the toxicity of micronanoplastics? So having this understanding how plastics can be transported, but also accumulating at the placenta, we think it's very important to look at two mechanisms. So not only studying particles can cross, and then by crossing to the fetal tissue, induce direct toxicity in the fetal tissues, but also looking at indirect effects. So even if these particles do not cross, they can indirectly harm, maybe by interfering with important placental function. And we have heard that they are can induce important signals for the fetus, like hormonal factors, vascular factors, inflammatory factors. Just this year, we were able to publish a study that this is true for nanoparticles in general. So this is not about the plastics, but it was we used these like source particle, titanium and silicon dioxide nanoparticles, where we exposed placental tissues to these particles. And we just wanted to see how does this interfere with all these communicating factors that are important for fetal development. And this was a secretomics approach, and we could see a lot of deregulation of several like inflammatory factor, vascular factors, hormones. And we were then also curious, is this change sufficient to induce something or toxicity in a fetus? And of course, we don't have a fetal model in vitro. So what we studied were some alternative models looking at angiogenesis in a headcam model or neurodevelopment with early neurosphere tissues. And just to summarize, we could see that just taking this condition supernatant and removing the particles, that these combined alterations can indeed impact the development of blood vessels. As you can see here, it's much more reduced, but didn't have so much effect on the neurodevelopment. Of course, this is no proof yet for a disease, but it's of concern because proper vascularization is important for fetal development, placental development, and pregnancy overall. So what about plastics coming back? So are these toxic to placental cells? So we used here a very simple model and used these cells where the plastics accumulate most. You can see micronanoplastics accumulating in these trophoblast cells. And what we could then use this model for is that over 48 hours of exposure, we didn't really find toxicity to these cells, even if we exposed to really high concentrations, which are at the moment probably not realistic. We also looked at barrier integrity, and we didn't see any effect here. But we were then interested, knowing that there is some toxicities from the animal studies, what could be potential toxicity mechanism, and we decided to go for a study on gene expression and look at the overall gene expression after exposure to plastic particles. So we exposed in the perfusion model, which is having all the cell types and at the highest complexity, we exposed for 6 hours to these polystyrene plastics, and then took the tissue for a gene expression analysis. What was very interesting and what we didn't really expect was that we find the most deregulated genes from the polystyrene exposure rather than from the more toxic control or the copper oxide particles that we introduced as a toxic control, so we would have expected more there. And when we went for deeper bioinformatics analysis, like looking what biological pathways are affected, I just give you some snapshots, like one pathway that was prominent was inflammatory responses, maybe not so unexpected, with a lot of genes deregulated have an important role in inflammation. We also looked on the pathway level, and the most deregulated pathway was related to mineral absorption with iron transporter proteins expressed, so indicating that polystyrene might have some effect or impair iron homeostasis. And this is interesting because it's already known from pathogen exposure, like when we are exposed to bacteria, this can induce an inflammation and also similar IL-6 upregulation, and then the cell try to compensate this stress by decreasing intracellular iron level, for instance, by upregulating lipocalin, which is a chelator of iron ions. So we checked this in our samples too, and we found, interestingly, we also had upregulation of IL-6, and we could show that we also get the upregulation of this iron chelator, and that the cell tries to respond then to import more irons and downregulating the export of ions. So there is some dysregulation of these iron homeostasis. We don't know yet what is the impact, but it's of course also a bit of concern because this is known that iron has important roles for successful pregnancy. So because this is an endocrine disruption symposium, so I also wanted to show this group of genes that we found deregulated, both from the copper and the polystyrene, and it contains a lot of hormones. So we also find deregulated gene expression of leptin, chorionic gonadotropin, and some other pregnancy-relevant hormones. So which could indicate maybe that there is some endocrine disrupting activities, but if these sustain or are strong enough to induce a response, needs to be followed up. Embryo toxicity. So here, as I said, we don't really have good models, but we teamed up early with the Fraunhofer Institute that had the mouse embryonic stem cell test, which of course has some drawbacks because it's based on mouse models and it's very simplified, but nonetheless, we could show that this is testing 3 endpoints: toxicity to normal fibroblasts, toxicity to the stem cells, and toxicity to their differentiation to beating cardiomyocytes. And both the polystyrene nano and microplastics classified as weakly embryotoxic. Interestingly, the micro-sized plastic were slightly more toxic than the nano-sized form. So in the last years, we teamed up with DTH Basel to develop a placenta embryo chip, so to also be able to look a bit more on the side what's happening in a developing embryo. So what we did here is on a chip, we co-culture an embryo model, a very simplified embryoid body, together with a placenta barrier under dynamic conditions, which is achieved by tilting of these chips. So a user-friendly platform. And the idea is that we can use this model to test not only direct effects when something can cross the barrier, but also these indirect effects because both organs are in close vicinity and you don't lose the short-lived intermediates like ROS species or maybe some inflammatory factors. We did one very preliminary study with microplastic particles. So these particles do not cross, as you can see here, the placenta barrier, but they accumulate in the placenta. And when we looked at toxicity, it was very interesting that we saw in the gray bars, you can see when we directly exposed the embryo bodies to the plastics, there was no decrease in the viability, so no direct toxicity. But when we added the placenta and the particles could not even reach the embryo, we saw some toxicity actually occurring, which could indicate maybe there is some indirect effects via the placenta. So which I think highlights that the placenta should really be an organ that is considered also in these NAM testing strategies. So my take-home messages here are, yes, micronanoplastics can cross a placenta barrier, not as readily as small drugs or molecules, but they can eventually, depending on the size mostly, but also on some other properties, so it's complex. And we should look not only in the direct mechanisms from the cross particles, but even if we don't see a crossing, we should maybe still consider indirect toxicity. And of course, we have some ideas on some mechanisms now, but this needs to be followed up, so we cannot say yet if this will turn out to a disease or if the organism is capable to respond or correct these changes in gene expression, for instance. And as a short outlook, so we are continuing and we are pushing this model because we see some value of it to be fully human-based system, because at the moment It was with mouse embryoid bodies, but we will move to an iPSC, human iPSC-derived embryoid body, and also like improving the placenta barrier with primary cells that form a true syncytium. We will now, most importantly, move away from these artificial plastic particles. I mean, they might be interesting for some answers, but we are most interested in what happens to the plastic particles we are exposed from the environment. And we try, and it will be very hard because it's very hard to mechanically grind them down to these small sizes. Also, we can mimic these environmental plastics with UV aging. I mean, it's just a surrogate, but we have shown in other studies that you can induce chemical and morphological changes just by UV treatment, which gets a bit more closely maybe your plastic model particles to the reality. Also, this UV treatment is inducing higher toxicity than the non-treated plastics, so we should really move towards more realistic plastics. And we are curious about what is the role of additives and contaminants. We have one newly started project. We are specifically interested in what is the role of microplastics to prime towards diseases in later life and immunological diseases, like maybe a role in allergies, or if they could co-shuttle some allergens from the environment towards the fetus. And also very excited that we got a new grant funded from the Horizon Europe for the next 5 years, which is exactly tackling the disadvantages of the NAMs. So we will approach from this artificial, maybe experimental side with our models on the mechanisms of toxicity, but in parallel, we will have a lot of people doing biomonitoring in 3 different countries and collecting birth cohorts and looking, of course, collecting samples that they are not contaminated with plastic. And maybe by bringing these 2 fields together, we might get closer to an understanding what is happening. So with this, I would like to thank my wonderful and highly motivated group, clinical scientific partners, funding, and thanks for listening.